My name is Luxin Wang. I'm assistant professor working for the food science department. My great pleasure to be here. This is my second time giving talks at this Postharvest fresh cut meeting. Great pleasure to be here. So today my title changed a little bit. Last year I talked about spring mixed salad. This year, since we got some good data about romaine lettuce, so I will give it to you. The experiment I will share with you is going to be the experiment we did with romaine lettuce. So that's why the title is Involvement of Microbiome Analysis in Food Safety Research. You probably start to hear people talking about microbiome analysis here and there. Many companies started doing that, and I want to show you how this microbiome analysis is going to help you, even though it has not been widely adopted or not really going to give you a direct answer, but we definitely see good potentials from those analysis, from those techniques, and the result we can obtain from these techniques. Based on years of research, we know that the level and the composition of the bacterial population present on those vegetable crops are going to be different, just like us, okay? Last year, we had probably 80 people here and different group of people, right? This year, we probably have 70 people here or 100 people here, different group of people, just like microbes, right? They're going to be different numbers and also different composition, different types of microbes. According to the previous research, we know that raw leafy green vegetables, if they are unwashed, they have about 8 log CFU per gram of bacteria on their surface. And then the field study has already looked into the diversity and levels of those microbiome present on those leafy greens. And this is the study conducted by Dr. Maria Marco from UC Davis. What they did is that they grow romaine lettuce in Salinas, both in the 2009 and 2010. Here's their result. When they have E09, which is the pie chart on the very left side, that means early season 2009. When they have L09, that is late season 2009. E10, that's early season 2010, and then the very last one is late season 2010. So what they see is there's 4 major bacteria phyla present on those romaine lettuce. Those are proteobacteria, which is the blue, dark blue, Firmicutes, which is the really dark red, green actinobacteria, and the purple Bacteroidetes. Once they collect those romaine lettuce leaves from the field, they analyze the composition of them. I mean, apparently for all the romaine lettuce, they harvest from those Salinas growing area. Among all those 4 times, those 4 bacteria phyla, they always present on the top. But the composition, the percentage of those bacteria phyla, they were different. As you can tell, for the early season, which I want you to focus on the first pie chart and the third pie chart, major part of the pie actually are Firmicutes, which is the dark red. When you look into the late season, you can see the majority of the pie chart is the dark blue, which proteobacteria, which shows that when you are harvesting those romaine lettuce at different season, you're going to see different composition of those bacteria phyla. From there, we know that after we harvest them, okay, we rely on those washing and sanitizing steps to control those microbes. Among all those 8 log, even more than 8 log bacteria cells, we need to remove them to extend their shelf life, protect our food quality, and also, most importantly, equally importantly, is the food safety, right? We add sanitizers in the wash tank in order to prevent cross-contamination. According to the research, approximately 90 to 99% of the bacteria can be removed. It's an impressive number. We say, oh, 99% is good. But then if we think about, we start at 8 log, which means that that's about 100 million cells, then 99% off, we still have about 6 log, which is about 1 million cells left on those romaine lettuce. So the question now we have is, well, what have been removed? right? I mean, I know there's a lot of them, and know there's different types of cells are there. What have been removed during the washing process? Because what we care is, I hope, it's not going to be those low-level E. coli 015787 or Salmonella or Listeria got left on that leaf and get people sick. Another thing is, we know that there's different between the lettuce we harvest from different seasons. After washing, will the seasonal differences we see on those harvested romaine lettuce remain the same after washing? Okay, what's going to happen? And how are they going to impact food safety? So that's the major question we want to address and for this particular project. And to make it easy, we went to the grocery store. We obtained 3 brands of romaine lettuce, chopped romaine lettuce products from the supermarket. After we obtain them, we get them in the lab and inoculate them with E. coli 015787 or Listeria monocytogenes. Those are pathogens that we work on. As you can tell, we inject, use a needle of those pathogens into those bags of salad, and then we'll seal the bag, and then we put it back in our refrigerator in the lab. We also sample the product after 5 days and to see if those microbiome going to impact the survival or potential growth of those pathogens. We also have a batch of product that we did not inoculate serve as our control. At each time point, we collect those romaine lettuce and we homogenize them to release the microbes from the leaf so that they get into the solution. So we can plate them on the plate, which is the top part. It's chrome agar, the one I'm showing you, it's chrome agar 015787 to help us enumerate the surviving 015787 cells. And then the bottom part, I want to tell you, we also extract the total DNA from those samples with the idea of understanding the diversity, the composition of the microbial populations in there. Because we want to answer the question after washing, were the seasonal differences we see remain the same, okay? So here's our result. This is the plate count result. We have 2 figures. On the very left side, this is the count for total aerobic bacteria, which means that those are the bacteria prefer oxygen to grow. On the left-hand side, those are total anaerobic bacteria, which means those are the bacteria that do not like to have oxygen. So from the left-hand side, we want to show you the comparison of 3 brands, A, B, C, and also the comparison of A, B, C, and comparison of early season and late season. One thing I want to point out is when we purchase those samples from the grocery store, even though we talk to the grocery store manager, we get those samples immediately after they got it almost, but still it's hard for us to control that they have exact the same expiration date, being to the exact same cold chain. There's no way. So when I talk with my graduate student, I say, how do we standardize that? I mean, one thing we can think about, and I think that's the best we can do, is we will plate all those samples on their use by date, best use by date, consider as our way of standardize among those 3 brands. Because we don't want to plate one sample that has 2 weeks before the best use by day, and the other sample has 5 days. That's not fair, right? So we all plate them on the same date, their best use by date. So that's something I want to mention. As you can see, for total aerobic plate count, we didn't see much difference among A, B, C brands. The only time we see a small difference, even I put 1 star on top, that means the p-value was 0.05. Well, it's not, it's significant, but the difference was not big. It's about 1 log difference between the early season A and late season A. On your left-hand side, which is the total anaerobic plate count, you can see we see more differences among 3 brands, the A, B, C. Apparently, C brand had the highest total anaerobic plate count compared to brand A and brand C. And also, we can see that brand A, again, their late season had the higher total anaerobic plate count compared to their early season. And brand B didn't change much, and brand C didn't change much. The 3 stars on top, or the 2 stars on top, represent different p-value, which means that that difference was more significant compared to the others. So again, this is just a count, okay? When you look into the plate, they all look like this, right? On your right-hand corner, you cannot tell where they are. So that's why we decided to do the sequencing and we want to do the microbiome analysis. Just a quick sentence here. Very limited information can be really obtained from your plating methods because only 0.1 to 1% of the total bacteria can be cultured by plating method, which means that what you see is only a small portion. In other words, 0.1 to 1% of the total bacteria population present in there. That's the part they can grow, develop into a colony, and you can now see them, okay? Majority of them, they will not grow, you cannot see them, okay? So that's why we rely on sequencing, which rely on their DNA information to tell us where they are. I want to quickly tell you how we do that. Because this is a picture on your right-hand side. You can see there's different types of microbes. It's a cartoon. I think it's very, it can help us vividly understand how do we understand their diversity and how do we understand their abundance. When we talk about microbiome analysis, we cannot, first of all, let's go through this picture. You can see we have different types of microbes. If I go over here, I hope this one can help me. Okay. For example, this one, right? This is the blue one with a red coat with dots in the middle. This one has 1, 2, 3, about 10 cells here. This is considered one type of bacteria, and this 10 cells is their abundance, right? They have 10 of this. If I look at this one, which is a very cute one with an orange core and a lot of red hair coming out, and this one, even though it's very unique, only have 1 cell, right? If I look into another one, this one, okay, it's like a long one, 2 pieces. Well, blue with orange hair coming out, so 2 copies of that. Okay, when I look into this picture, I want to know the diversity. I extract the DNA. So what I will do is I will amplify those DNA and I will blast those DNA information into a DNA information bank. So as long as they match, then they will tell me, you know what, Lucian, this one is the blue one with it's DNA, but they will tell me, Lucian, the blue one with the yellow hair is there. I blast again, it will tell me, Lucian, this one with blue and red coat is there. It will tell me what they are. When they try to tell me how many of them are there, think about it. They are very small and they only have 10 cells, 1 cell. It's hard to really count them. So what they do is they call relative abundance. In other words, they tell you the percentage. I assume this is 100 cells in there. In the end, they will tell me, this is 10, right? They will say 10% of them is going to be this blue one with the red coat. 1% is this one, the yellow one, orange one with the red thing, and also you probably have 5 of the sausage type thing, like orange with blue, 5% of them. I assume the total is 100. So the information you get in the end is relative abundance, okay? But it can tell you overall picture of this, what type of bacteria there, and what is percentage of each bacteria. Okay, that's the information you will get. Based on this diversity, how many and what type of them, and their relative abundance, we are able to compare those different brands and different early or late season products. So this is what we get. Okay, based on all the information, right now, just think about it. We already massaged all the diversity and also the abundance information together. You can see that the A product, based on the diversity and the relative abundance of those bacterial populations, they kind of group together. This is B, the red one is B. The A product, they kind of grouped together, and the C, they again grouped together, okay? And then on the right-hand side, these are seasons. We can see our product. If we look into seasons, all the early season, they grouped together, and the late seasons, they grouped together. Now, at this point, we still cannot tell, okay, what other bacteria that really are different. And here's the result we can get based on their sequencing. Again, we know brand, we know season, they can all separate our products. Now this has come to the relative abundance of those different types of bacteria. If you remember the first one, the one, I give it to you here, right? So Proteobacteria, Firmicutes, Actinobacteria, Bacteroidetes, those are the top 4 we identified when we harvest from the field. They were not washed. So now go back to this one, we can see that Proteobacteria, Firmicutes, Bacteroidetes, and Actinobacteria remain the top 4 bacteria after washing, okay? And then when we were looking to their relative abundance, that's why you can see on the y-axis, that's a relative abundance, the percentage. That's why I don't see abundance or concentration, I see relative abundance. Okay, so the relative abundance for Proteobacteria is the highest, followed by Firmicutes, Proteobacteria, Actinobacteria. Also, what we see is the 2:2 stars, Firmicutes and Actinobacteria, their relative abundance was significantly different among 3 brands. For Firmicutes, their relative abundance is higher in brand C, followed by brand A and B. For Actinobacteria, their relative abundance is highest in brand A, followed by brand C and B. Now the next thing you're going to ask me, and it's the question we asked before, how about the seasons? After washing, we know before washing, if you still remember this one, Firmicutes were generally most abundant in the early season. Proteobacteria is more late season. Okay, go back here. We realize, you know what, after washing, the differences we see about Firmicutes between early and late season no longer exist based on the comparison of their relative abundance. Wow, sorry for that. And then Proteobacteria, their differences still exist, which means that washing really kind of altered somehow, right? The Firmicutes balanced it out a little bit, where Proteobacteria, because of the high relative abundance, it didn't get much changed. And we see Proteobacteria, the late season is more than the early season. Okay, the next question we want to answer is, we see the difference, right? The washing did not change the relative abundance of Proteobacteria much between early season, late season, and we see that it changed the Firmicutes a little bit. And, okay, Lucian, tell me the thing here. Are they going to impact our pathogens? So what we did, as I mentioned earlier, we inoculate those samples with E. coli 015787, and also another set of samples that we inoculate with Listeria monocytogenes. This is our data for the E. coli 015787. We inoculate those samples on the day that they have this used by date, same time for among all 3 brands. We see that E. coli 015787 did not grow in many of them, okay? Basically, they kind of stayed at the same level, and we put them in the refrigerator for 5 days, so there's no difference. And the next thing is, how about our Listeria monocytogenes? Listeria monocytogenes is known for their capability to grow at refrigerated temperature. For E. coli, it does not, okay? I mean, when I see that data, I won't say that it's really the microbiome's contribution. I mean, because E. coli, naturally, they just do not proliferate under the refrigerated condition, but Listeria does. So we see that for this particular B brand, we have it in the early season. This brand supported the growth of Listeria monocytogenes. And nothing happened for the late season product, only this B brand in early season product. Here I want to highlight one thing. The reason I always choose 3 brands, or sometimes we even choose 5 brands, depend if we can consistently get that or not. Its reason is we know that different processing plant, you guys use different washing strategies, double wash, triple wash, or different solutions, PAA, chlorine wash. So that's why we want to have different brands so that we diversify. And when we give you the conclusion, it's applied to not only one product, at least we tested multiple products. So we see that it's only happening in brand B early season. So the next question we want to know, what is the difference between those microbiome populations in the brand B that really potentially contributed to the growth of Listeria monocytogenes? So here's our data. On the left-hand side,
I hope between minutes I know it's really 10 minutes.
Oh, okay, that's good. I'm like, how can I take up so many minutes? I'm like, I practiced once at home. So. I'm like, yeah, I shouldn't go that. Okay, good to know. I still have time. So on the left-hand side, that's the control sample, which means they are not inoculated. On your right-hand side, those are samples inoculated with Listeria monocytogenes. You can see that after inoculation, the Firmicutes concentration go up significantly, right? And when we do this comparison, we can see that the green box is inoculated, red box is the non-inoculated, and the green box here it shows the whether their relative abundance significant change or not. So for the inoculated samples, Firmicutes significant change, increase. For the non-inoculated samples, proteobacteria is higher. So what are those significant increased Firmicutes? And when we look into those bacteria into their genus level, we can see that the one that significantly increased is this gray bar, and that is Listeria. So we are able to use our sequencing to identify the ones we put in there so that we know, okay, that is right, because we put Listeria in there, so compared to those non-inoculated samples, the Listeria relative abundance now increased. Look at those gray bars over here, gray bars over here, gray bars over here. Again, we also know that it's not always going to be, the relative abundance is not going to be always the same across all the samples because we have early season, late season, and we know that the early season supported the growth of Listeria monocytogenes. So then the next thing we decide to do is, okay, because we know the early season is the one really supported Listeria mono growth. So the next thing we want to do is, let's look into the non-inoculated salad, brand B, compare their early season versus the late season. That's, again, looking to the inoculated brand B, the early season, the late season. We want to identify what is the bacteria that remain the same or high abundance. And maybe that's the one for example, in the late season, didn't support the growth of E. coli 157, Listeria mono, is the one that really going to inhibit their growth. The ones that maybe in higher abundance in the early season remain in the higher abundance after inoculation may be the one that could support the Listeria mono growth, right? Because we see that there's a difference between early season, late season. So that's what we want to find out. What is in the early season, late season, non-inoculated samples? What are the top bacteria phylum or genera? So what we see is for the late season, Pseudomonas, which is a spoilage microorganism, is the highest abundance bacteria genus. And for the early season, we see another 5 of them, including Flavum bacterium, Nasomonas, Leuconostoc, and some others. So that's the 5 that's in higher relative abundance in the early season compared to late season. And then if we look into the one that inoculated with Listeria mono, for the late season, Pseudomonas still remain at higher relative abundance compared to the early season, and Actinobacteria is also now higher in the late season inoculated salad samples. And for the early season inoculated salad samples, we see another 6 bacteria genera that is higher relative abundance in the early season compared to the late season. So if we combine these two figures, now we can see for Pseudomonas, we know that late season samples did not support the Listeria mono's growth. So we can think now probably the high relative abundance of Pseudomonas in the late season samples might be one reason that inhibits the Listeria growth, because this one was higher in their relative abundance before inoculation, their relative abundance still remain higher after inoculation. So that we think that potentially be the one that, because they are high in there, suppressed Listeria mono's potential growth. Well, the others, like the one we call Xanthomonas, their relative abundance was higher before inoculation in the early samples, and their relative abundance will remain higher in the inoculated samples, also in the early samples. And this might be the bacteria genus that probably supported the growth of Listeria mono. Okay, of course, this is just based on those bacteria, phylum, bacteria genus, the sequencing analysis, and it can at least help us to start to think what are the potential bacteria genus, bacteria species even, in there that need to be better controlled in order to prevent that potential growth we see over here. That's what we don't want, right? And the next thing we start to think about is, if we know the new target, and we do know that different washing sanitizers have different target microorganisms, then based on that information, for the early, for the late season, we may want to be able to develop new strategies to control the ones that is the problematic species so that they won't cause growth. Those are all ongoing research. We just want to let you know what we find, and of course there's additional things that we need to do in order to confirm, hey, is it really Pseudomonas, or probably we need to do trials like different washing solution and or artificially inoculate the samples with more Pseudomonas to see if that really the truth. A lot of things need to be done, but at least we want to show you that's how those microbiome analysis, layer by layer, can tell us those information, can help us better design our study, can help us evaluate our washing efficacy, and can help us really identify new targets, right? So that's what I want to present, and thank you very much.